So welcome to everybody to this book festival virtual evening at Tulane University. I hope you're all well. I know that's a colloquialism these time of years, but this time in our existence, we mean it quite seriously. I hope you're well. And in light of the vaccines and the improving weather and the increased optimism, I hope you will enjoy this evening an extended discussion about, in a sense, some of the most important issues related to the pandemic and how we're coming out of the pandemic and how it's changing the world. I'm quite excited about this. First of all, because it's virtual, we can bring in individuals from across the country and the world to participate in this, the New Orleans Book Festival. And then secondly, the topic, as I said, is so important given what's going on in the world right now with the pandemic and how we're coming out of the pandemic. And then finally, I think it gives you a preview of the New Orleans Book Festival, which will be this October, not virtual, but in person, and you'll get a sense of how exciting this is going to be. Now, of course, we have an interview with the truly incomparable Walter Isaacson, who is a biographer extraordinaire, and as we all know, has written biographies, classics all, about some of the most significant figures in US and world history. Leonardo da Vinci, Stephen Jobs, Albert Einstein, Henry Kissinger, and in every case, it's a story about an individual and how they changed the world, but the circumstances that led to that, and then finally the implications for the world of in a sense, the innovations that they brought about. And of course, the book we're going to speak with Walter about today, The Code Breaker, Jennifer Doudna, Gene Editing, and the Future of the Human Race. I think that says it all about this biography. Jennifer Doudna, of course, was the developer of CRISPR technology, which brought global acclaim as well as the Nobel Prize, and raises a host of issues about how we, in a sense, might prevent future pandemics through screening and treatments, but more importantly, how we might, in a sense, engineer human beings so they might not be subject to those diseases. And of course, the bioethics issues that surround all of these different advances. It's a topic that has particular resonance at Tulane, as everyone knows. Tulane was founded fighting a pandemic in 1834. So infectious disease and biomedical research, issues of health equity have been at the core of the institution. And then over the last year, Tulane has really been in the forefront of infectious diseases, working on vaccines and treatments related to the pandemic. And we think of ourselves, given the explosion that in research has occurred at Tulane, is looking forward to becoming the biomedical bayou here in New Orleans in years to come. And in light of all that, there's not a better person to be an interlocutor for Walter Isaacson this evening, somebody who's been on the vanguard of this biomedical revolution, Trivia Frazier. She's president and CEO of Obatala Science, which is doing path-breaking research on regenerative medicine and advanced cellular engineering. I have to say, we claim her at Tulane. She has 3 Tulane degrees. She was in the joint program in physics and biomedical sciences with Dillard as an undergrad, and then got graduate degrees at Tulane Medical School and the A.B. Freeman School of Business. She's run an incredibly innovative, diverse company, and clearly is in the forefront of the issues that we're going to talk about this evening. So with that, I want to turn the virtual mic over to Trivia Frazier.
Thank you so very much, President Fitz. What an outstanding introduction. We are really just thrilled to have this discussion and, to kick off such a timely discussion, I think, is what brings me the most joy to moderate a discussion with Walter, with some of your very own work. I think, in terms of what this means for not only the local area, but, in terms of generations to come, we'll see this book being a reference with what's going on, what happened during the COVID-19 pandemic. And so with that, I'll start off by just giving an overview of, what our company does in terms of biotechnology and what it means as a mean of providing toolkits, like what is CRISPR? And then Walter wants to give a special shout out to someone, and we're looking forward to that. But essentially, as President Fitz mentioned, Obatala Sciences, I have the immense joy of being at the helm of a biotechnology toolkit company. We essentially manufacture and assemble biological materials to mimic tissue from patients of varying demographics. The idea is to support safer and more efficacious therapies for the treatment of illnesses such as obesity, diabetes, and regenerative medicine. We're developing this technology using stem cells, and the stem cells have been used very broadly as a means for administering certain types of therapies, investigating how to do that in the context of certain diseases that are really impactful around the globe. But there are many other types of tools. And one of those is the CRISPR-Cas technology, which you hear in a term of CRISPR, very commonly used. Walter has been a champion for taking us through some of the significant time courses in our lives when such significant discoveries as CRISPR have been made. And so today we'll discuss one of the pioneers in this technology. Dr. Doudna will walk through some of the highlights of the book and hopefully share just enough information that you'll want to go out and buy the book. And so before we jump in, we'll see a very special person that I mentioned that Walter wants to give a shout out to.
Well, somebody who works for you, Trivia, Spencer Olewski, I don't know if we can pull him up, but he was Spencer. He was a scientific veteran for my book. He took my undergraduate course at Tulane, majored in the life sciences, but he was one of two people who did the scientific vetting of the book. And now he's just been admitted to Tulane Medical School. And so he's going to be a superstar in this firmament of the New Orleans. I love the biotech bayou idea. In this firmament we're going to have of entrepreneurs meeting the life sciences and fulfilling Tulane's mission ever since the 1830s of finding ways to help humanity. And Spencer, you also broke your foot, which I think is kind of a neat way to start this. So thank you. Say hello to your boss, Trivia, and to your former professor.
Yeah. Thanks for the shout out. I appreciate it. Yeah, no, it's such an honor. It was such a great opportunity and I was able to learn so much. So incredibly grateful for this.
Well, you were great in helping march me through the basic biology of the book. So thanks. Listen in. If you have a question, raise your hand. Maybe Trivia will call on you. And I'll turn it back to you, Trivia.
Thanks, Walter. Good to see you, Spencer. Great segue. And this story essentially is about a researcher, a Nobel Prize winner, a public policy thinker, but it's so much more than that. So we're going to jump in. But before we do that, I'll do a terminology check. All right. So as I mentioned, you hear us use the term CRISPR. It literally stands for clustered regular interspace palindromic repeats. Okay, that's a lot. What's a palindrome? Quick sort of reminder, a palindrome is essentially a word, phrase, number, or sequence that essentially reads the same backwards as forward, like the words civic, radar. I'll use the number sequence 12121. And essentially, it's a gene editing technology that is based on a trick, an old age trick that bacteria have been using for billions of years to protect themselves from viruses. In their DNA, bacteria take sequences and they basically steal them from viruses that have attacked them in the past. If they survive that attack, they now have a memory of that specific virus based on that unique sequence that that virus left behind that is now in its genome, its DNA. And so it's essentially, I'll use a reference, something that is more easily recognized, a miniature Pac-Man. And we may refer to that term a little later, but if you can envision a Pac-Man that can pick up some very unique sequences or alphabets or numbers, it searches the DNA. And when it sees a sequence that doesn't belong there, so if we have ABC 12121, DEF, it goes in and it takes out that specific 12121, that sequence that doesn't belong. And in a way, that is an innate an adaptive immune system for the bacteria to thrive, to survive another attack. And so Walter basically takes us through this technology, the significance of it, how Jennifer Doudna actually started and embarked on this particular journey, taking us from even the 6th grade when she was first introduced to the concept, right?
Right.
And so Walter, you talk about there are 2 themes in the book. You reference how something works, especially when that something is ourselves, and a simple phrase that nature is beautiful. Could you share how those themes come through in the book?
All right, but first, let me talk about the name CRISPR, because you're about the only interviewer who actually knew what all those letters meant. But one of my favorite little tales in this book is this guy, Francisco Mojica, a graduate student in Spain, who first saw these in the 1990s and these bacteria found in salt ponds. And they named them things like sequential repeated sequences, whatever, or tandem repeats. And none of the names sounded very good. And so one day he was trying to figure out, if I can come up with a clever name, I'll go down in history. And so he thought of the name CRISPR and then reverse engineered it so he could have all of those words that spelled out CRISPR. And his wife said it sounded really bad. It sounded like the name of a puppy dog, but it ended up catching on. So that's how we get the name CRISPR. But as you said, it's simply a system bacteria have used for 1 billion years to remember viruses that have attacked them and fight them off the next time, which I don't have to tell anybody listening, is a kind of useful talent to have for us humans in this day and age. Jennifer Doudna is the heroine of this book. And as you said, when she was in 6th grade, her dad left on her bed the double helix. You and I remember reading that when we were young, I'm sure, is Jim Watson's accounts of how he got the structure of DNA. And she thought it was a detective tale and she put it aside. But when she read it on a rainy Saturday, she realized, well, it actually is a detective tale. It's a detective's tale looking at the clues for the secret of life. But what struck her was there was a character in the book who's really interesting, but treated in a condescending way, named Rosalind Franklin, who did the images that helped Watson and Crick do it. And Jennifer said to herself, wow, I didn't know girls could become scientists. And that's another reason I wanted to write this book, is you need role models in life. And she found one in Rosalind Franklin, so I hope she can be one to a next generation of girls as well as boys who say, I want to grow up and be a scientist. And so she goes, and while all the guys are trying to figure out DNA, she studies RNA, which turns out to be so much more interesting as a molecule. She discovers how it can replicate itself, which means that it was a molecule that probably began life on this planet. When she studies CRISPR, she realizes that RNA is the guide that takes the scissors and cuts the DNA, like you talked about. And then, of course, those of us who have been jabbed with the Pfizer or Moderna vaccines realize now how RNA can be its basic function in the planet is being a messenger, taking the code from the DNA that's in the nucleus of our cells and bringing it to that region of our cells where we make proteins and telling us to make a fragment of the spike protein on the coronavirus. So boom, just like bacteria, if they see that mugshot, they know how to kill it. Now our body sees that mugshot and knows how to kill it. So I started this book thinking that the biosciences was the next great revolution, that it was filled with colorful characters, that it was an exciting detective story. And then when coronavirus hit and she won the Nobel Prize, I realized, hey, I was understating the case. It's even more exciting than that.
Absolutely. Outstanding. And you talk about, one of the things that came across Doudna as sort of a revelation is hearing and finding out that women can be scientists. That actually is great because the first question that I have, or at least I would say secondary question, you did a great summary of the book, is more so, that introduction to Rosalind and, Rosalind Franklin's story and, reading about it and what happened there? Why was that so impactful for Dr. Doudna?
Well, Jim Watson, in writing the book, calls her Rosie, which she never called herself, and talks about her looks and treats her a bit condescendingly. But to give Watson a little bit of his due, if you read the book carefully, he smuggles in a large dollop of science, including a deep respect for crystallography that Rosalind Franklin is able to do. And as you know, crystallography is just a way to do images of tiny molecules by seeing how it scatters the light, and you make images or photographs of them, which is what helped Watson and Crick figure out the structure of DNA. And so I think she developed this, was very impressed by this, not only the notion that a woman could do science, but in a detective story about the secrets of life, it's not just about the chemicals or about the molecules or even the coding of RNA and DNA. It's also about the structure. You gotta figure out how does it twist and fold to figure out how a molecule can connect with others or do things. And so that puts her on the path to become a biologist who cares about structure, who cares about biochemistry, and is eventually able to use that to figure out how does the CRISPR system work? How can we make it a gene editing tool? And then when coronavirus strikes, how can we take RNA in general and make it a tool that we can use to fight viruses?
Wow, just amazing. I mean, the detective's tale, that tone, it does bring excitement and wonderment for a child, for, an adult as well. One of the other things that was very significant, and it sort of feeds into, the idea of Jennifer Doudna sort of going down this detective's path. But it's something that is also a clear tone throughout the book, and that is the idea of collaboration and teamwork within the sciences. You talk about it also being, a concept that's prevalent within the book as well, writing a book. Could you give more, information about that? How was collaboration important for Jennifer? And, of course, it talks about some of her colleagues, starting off with Emmanuel Charpentier, or is it Stramanti, in terms of the reference, having that charisma, being someone that she was able to work with and develop such a strong relationship with.
Yeah.
Could you speak to that?
Innovation is a collaborative effort. We all know that from life. And creativity is a team sport. And sometimes we biographers distort history a bit and make it seem like some guy or gal goes into a garage or a garret and has a light bulb moment and innovation happens. And we all know it happens instead because people build on each other's work, form teams. I hope President Fitz isn't still listening. I know Brian Edwards is, and he's the dean. So I'll say this in the cone of silence of this thing. One of the things I do in the class I teach at Tulane, and Spencer, I think, remembers that, is when you do your project, I say you can work with other people. You can form a team to do it. And people say, well, how are you going to do the grading? How are you going to know what it is? And I say, Well, when I was young, they had another name for collaboration. They called it cheating. But I really think you're going to learn more if you learn to collaborate with your fellow students and form teams to do the project. Well, Jennifer is spectacular as a team builder. She does a type of team building that is interesting because you can argue it either way. I remember talking to Doris Kearns Goodwin about it because she wrote a book, Team of Rivals, about Lincoln having clashing members of his cabinet, and about Franklin Roosevelt, who liked creative tension. Jennifer's the opposite. She makes everybody who's going to be in her lab or she's going to hire or have as a postdoc. She says, I want you to meet everybody in the lab, and then they all meet to see if that person will fit in and get along. And I said, well, don't you lose something by not having the round peg in the square hole, as Steve Jobs would say, the person who's going to challenge everybody and maybe get on their nerves. And she said, that's a good way to do teamwork and collaboration, but also my way is a good way, and it fits me better. So Trivia, I know as you form the team at Obatala, you have to balance that. Do we want creative tension, and do we want pure collaboration and teamwork? Certainly it pays off for Jennifer. She's got the most loyal team of people, but being a person who likes collaboration and cooperation, she's tough. And I mean that as a high compliment. She's competitive. And when she got into a competition with a group of guys at the Broad Institute of MIT and Harvard, she didn't give much quarter. She raced them in order to be the first to publish on CRISPR-Cas systems. She's fought them on the patents for use of the gene editing tool. And so in life, and almost everybody I've seen on the list of viewers here knows that you have to compete sometimes and you got to cooperate sometimes. And both are valuable, including competition, because it spurred her and her partner, Emmanuel Charpentier, to stay up all night, stay up on weekends. Emmanuel was in Europe. Jennifer was in California. They'd sort of hand off at when it was night for one person, it was morning for the next. And that competition, just like the competition of Watson and Crick competing against Linus Pauling to find the structure of DNA and against Maurice Wilkins and Rosalind Franklin, spurs people to go faster. So this book is about how creativity happens. It's not just about biotech, and it's about how competition as well as cooperation are key to making innovation work.
Yeah, yes, absolutely. And as I understand, she had some very, very tough competition at MIT. Feng Zhang, you write about, interacting with her competition, who actually was good to speak with you and was actually open to that conversation. So,
Feng Zhang, who was her main competitor at MIT, I think the only thing that Jennifer Doudna, who's read this book, I spoke to her again this morning, the only thing she doesn't like about this book is Feng Zhang comes off pretty well. I kind of liked Feng Zhang, even though he was the rival. He was born in China. He's much younger. He's in his 30s. But when he was a young kid, he gets his mom brings him, and they move to Iowa. And they grow up in Iowa, where he's just embraced in the community there, the way the heartland can do for immigrants. And he becomes this cheerful, optimistic American who just aces both computer science and biological and genetic coding as well, and becomes this superstar at Harvard, MIT, and the Broad. But he does get into this race and is still in this patent battle with Jennifer Doudna. And he knew I was writing about Jennifer Doudna, but when I went to Kendall Square in Cambridge, we had lunch at Legal Seafood. We had dinner together. I met all of his lab. I worked with him. And so he was very generous with his time. And I guess that may be why he comes off well in the book.
right. But so despite those differences, he was able to work towards using CRISPR in the fight against COVID-19 as well.
That was interesting because their competition is really bitter and their patent fight is even worse. Yet, exactly a year ago, at the beginning of March when the shutdown happened, Jennifer Doudna is has sent her kid, Andy, a 17-year-old boy, to Fresno, California to be in a robotics competition. And Jennifer wakes her husband, Jamie, up at 2 a.m. and says, we got to go retrieve him. This pandemic is worse than I thought. I don't want him at this robot competition. He drags the only child. And so Andy is like rolling his eyes. But then they announce robotics competition canceled. And Jennifer says, okay, the next day she gathers 50 scientists and said, how are we going to turn our attention now to using CRISPR as a testing technology? How are we going to turn it into maybe an antiviral technology? How are we going to set up labs and help fight this? And across the country, Feng Zhang was getting emails in Chinese, even though he, didn't live in China. The Chinese embassy was saying, you have to help us here. When one place is in trouble, help comes from all quarters, an old Chinese saying. And so he does the same thing. He gathers a group of people at the Broad Institute to turn their technology to fight this pandemic. And this time, even though they're racing to publish papers and racing to make discoveries against each other, they're publishing them on the internet in the public domain instantly. And they're saying, we're not asserting any patent rights. Anybody who wants to use these new discoveries we're making each day to fight COVID is free to do it. So I think it turned their mindset to collaboration rather than competition, and reminded them and the rest of us that the real reason you, Trivia, and Feng Zhang and Jennifer and all are in this business, and Spencer will be, is not simply to win patents, not simply to win prizes, but because you want to do something that's going to make the world a better place.
That's right. That's right, Walter. Greatly stated. I mean, if I didn't know better, I'd say you were a researcher there.
I went into her lab and I made her graduate students teach me how to edit human genes. So I love the bench work.
That's awesome. It's amazing how much you've absorbed in this process. But
It's a lot easier, let me tell you, than general relativity was when I was writing about Einstein.
Speaking of that, and I would like to open up, to questions from the audience. But speaking of that, you also took us through what could be coined as the third technological revolution. And what is that? I mean, I understand it. And it's very exciting to think about it and to be in the middle of it, to see it happening, to see the shift in the world. But could you talk about that briefly?
Yeah, the third biotech revolution is you, and it's Tulane, and it's what we're all going to be doing, and what Fitz has now dubbed the biotech bayou, because the first great innovation revolution of modern times was, I think, the physics revolution. And it comes from the discovery of that fundamental kernel called the atom, and Einstein's theories about what the atom does inside and out, and how equals mc squared. And that brings us the atom bomb and space travel and GPS and semiconductors. Second great revolution also comes from a tiny kernel we discover, which is the bit, which is that every piece of information can be encoded in binary digits known as bits. That brings us the microchip, the internet, the computer. And when those three come together, you get the digital revolution that I wrote about with Steve Jobs and the innovators. Now we're entering the third great innovation revolution of our time, in which molecules are the new microchip. And kids who learn how to code digitally are also going to know how to understand the code of life and be able to figure out, how do I connect computer coding, the humanities, and the code of life? Because that's what we're doing in the third great revolution. I do think that Tulane, and I do think that New Orleans is very well positioned, because the digital revolution was all done by engineers, mainly in places like Silicon Valley. But here, it's places that have schools of public health, have medical schools, have primate centers, have great research universities that start an entrepreneurial ecosystem that produces things like your company that you founded, Obatala. And this new revolution in the life sciences is going to need creative people. It is going to need the type of innovation zones that Tulane, I hope, will be creating, so that we'll have a dozen more Obatalas. I hope none competing directly with you, but in sort of doing the innovation in this third great industrial revolution, which won't take place in Silicon Valley. It's taking place in places like Austin and Nashville and New Orleans and Boston, of course, where Kendall Square is a new epicenter. But it's places that have research universities, hospitals, and have a heritage of looking at things like infectious diseases and tropical medicine.
Right, right. Spoken so well, Walter. And so before I could ask you a slew of questions because this is such an awesome book. Let's open the floor up and see what questions we have. And we can start off with Spencer. Go ahead, Spencer, and we can see what your question is. Able to unmute yourself.
Hey, how's it going? Sorry, I think I had some mic difficulties. So I'm reading some of the questions, and I guess I'd like to hear Walter talk about the book process and how he went with the interviews and how he decided to approach, interviewing Dr. Doudna versus Feng Zhang, and kind of the narrative that he was looking to get to.
Well, one of the things I bring to a book like this, I'm not like you, Spencer. I'm not a superstar in biology or like trivia, but I'm a good old-fashioned reporter. I can call people up. I'm not shy. I can say, "Can I come on over? Let me hang in your lab," and I'll listen to them and talk. So I spent a lot of time in Berkeley, staying with Michael Lewis, another New Orleans friend here, who has a house right next to the Berkeley campus, and getting her to talk. And likewise, when I called Feng Zhang and George Church, it's like, yeah, come on over, come to my lab. So unlike doing a Leonardo da Vinci, where you're just reading the notebooks, I'm learning in real time, which is why the book can kind of be an adventure story, where it's a journey of discovery, almost like an odyssey, where I started at the beginning with Jennifer going on a journey of discovery, but in a certain point in the book, I'm by her side and we're both going on this journey of discovery in real time, and particularly in real time because the coronavirus hit after I've been 3 or 4 years into working on this book. So we both go into that in real time. And a small little aside because of that, it was interesting because it's obviously the first time I've reported a book during a coronavirus lockdown, but I got to be in all of her Zoom meetings in real time. She allowed me to lurk in her Slack channels. And so I'm watching in real time as they're sharing detection technology discoveries about using CRISPR to find the coronavirus. And likewise, I was allowed on the Zoom meetings of Feng Zhang and his company, Sherlock, which is the CRISPR detection company he founded in Boston. So it almost helped me that in real time, I could be reporting this. And what I do is my late great editor, Alice Mayhew, taught me is, she said, all things in good time. Don't flash forward, keep it chronological. Because if you keep it chronological, the reader is learning and you're learning things as the people in the book are learning them. And it helps build this body of knowledge. So we're all going on a journey of discovery together.
Mm-hmm. Awesome. And some of those elements are applied to our process.
By the way, I'm looking at a few things in the chat room, if you don't mind me picking up a couple of them. One is Jeff Balkin, who I know has been interested. I didn't realize that he is related to Sidney Brenner, who won the Nobel Prize in 2002, who worked with Crick and Watson. But Sidney Brenner was also an incredibly close friend of Jim Watson and was part of the process of figuring out, do we use genetic engineering and how do we make it safe? And there's a picture of him in the book, even though I don't discuss fully, as Jeffrey asked whether I do, the discoveries of Sidney Brenner. I certainly am a fan of his.
Awesome. We also have a question from Diego, Diego Rose. Can you talk a bit about your process and how much time you spent with, spent
Well, I
introducing
did
them?
talk just
Yeah, you
now about it
did.
in terms of Spencer,
Yeah.
but the one thing you asked is, yeah, I mean, I spent a lot of time and, lots of phone calls, whatever. I did record the interviews, and Kathy, my wife, who reads, the book and talks it through with me at all times. At the beginning, she was helping me transcribe some of my interviews, but I think she celebrated when I discovered, I can't remember the name of it, but a digital transcription that can actually now do voice recognition well enough that it could transcribe the interviews.
Wow. Okay. Let's see. We have another question from, let's look at Jeff Kessler's question. This is a great question. What is the relationship between a CRISPR technology and the development of the Moderna and Pfizer vaccines?
I sometimes fear I'm making it sound like they're very, very much the same, and they aren't. The Moderna and Pfizer vaccines use some discoveries, including ones that Jennifer Doudna made, about what's called messenger RNA. As I say, Jennifer Doudna helped figure out the structure of RNA and how it had many uses. And in the vaccine, it's the most basic use, which, by the way, Sidney Brenner, who I just mentioned, this great scientist in the 60s and 70s, helped figure out what messenger RNA is, which simply means it's the molecule that takes the code from inside the nucleus of our cell and acts as a messenger, bringing it to the manufacturing region of our cell in order to build the protein we want. So that's what messenger RNA does. CRISPR has another use for this miracle molecule that's the co-star or best supporting molecule in my book, and that's called guide RNA, which is RNA, as you so well described, Trivia. RNA can guide an enzyme or a scissors to a particular spot in our genome and say, cut it here, snip it right there. And so what Jennifer did that won her the Nobel Prize is not only figure out how this guide RNA works with the enzyme and scissors, but how to re-engineer it. So you can make a single guide and code it to cut where you want. But in both cases, it's really the basic science is that molecules, as I mentioned, are the new microchip, which means we can reprogram them. We can reprogram the cutting mechanism so that we can change which genes get edited. And we can reprogram the building of proteins mechanism so that when the coronavirus mutates and has a new variant, so far Pfizer and Moderna vaccines are fine with those variants, but if they mutate a bit more, we'll just recode that messenger RNA and say, all right, build a slightly different version of the spike protein fragment.
Right, right. Yeah, and related to that, we have a question from Charles Zena. And his question is, using CRISPR to eliminate disease states is quite appealing. Designer babies is not.
Yeah,
Or monitor and regulate ethical use of this technology. I think this is a great question because Jennifer Doudna joined in that charge, right? Of essentially.
Yeah, a large portion of the book, about a quarter of the book, is not answering that question, but marching hand in hand with Jennifer and so many other ethicists, scientists, and religious leaders as we form these questions and figure out what do we think about them. Shortly after she developed the CRISPR editing tool, Jennifer had a nightmare. And that nightmare was that somebody wanted to learn about it. And she walks into the room and that person looks up and it's Hitler. If you're hearing the rumbling here, we're having a nice thunderstorm down in the French quarters. I don't know if it's raining uptown.
That's a tone for that nightmare.
Yeah, exactly. And so what Jennifer does is she says, this will be bad if it falls into the wrong hands. We'll have eugenics, we'll have designer babies, we'll have people creating soldiers that, don't feel pain. And so she starts a process that's described in the book that builds on the process that Sidney Brenner and Paul Berg and others started in the 70s, which is scientists getting together with ethicists and saying, what rules of the road should we have? What guidelines should we have? Now, I think it's a problem if we start making designer babies and editing out the diversity of our species and allowing rich people to buy better genes for their children than poor, less privileged families. But I also think it's a problem if we get so frightened by gene editing that we don't do things like last year, sickle cell anemia was cured for the first time in a patient down in Mississippi by editing her stem cells. Likewise, Tay-Sachs, Huntington's, muscular dystrophy. And Jennifer told me that she started becoming more open to the use of this technology, even when it comes to making designer babies, when it comes to making sure that the babies don't have these debilitating genetic disorders. And even today, when I was doing a talk and somebody was saying, isn't this horrible? We're going to have designer babies. In the chat room, there were 3 people who put things up saying, I don't want to say this out loud, but my 12-year-old granddaughter is probably going to die in 3 years because she's got this genetic disorder. Can you put me in touch with Jennifer Doudna and maybe she can save her? And when you read, and there were 2 others like that, when you read those things, you realize it's not a simple question of saying, well, let's not do things that might save our babies or even edit genetic diseases like Huntington's out of the human species. Let's use this carefully. Let's go step by step, but let's keep our minds open to the beauty that science can do. Every creature on this planet, large and small, uses whatever tricks it can come up with in order to thrive and survive. And just like Obatala on a chip does human, cells so that we can thrive and survive, I think that's what we should do with this CRISPR technology, while also saying, here's some guidelines. No inheritable edits except for medically necessary treatments of major disorders. And that'll last us for about 20 years. And then our children can take over figuring out what the rules should be.
Right. And Jennifer was very clear about not promoting rogue therapies, things that are even, borderline unethical, but mainly speaking to what you're highlighting, and that is in the case by which we can save lives.
And Francis Collins, the head of the NIH, who I've talked to quite a bit for this book, the NIH and the FDA can create guidelines that make it very difficult for people to do things that haven't been approved. But more importantly, the Chinese scientists who 2 years ago created designer babies by editing early stage embryos has been arrested and put in jail by the Chinese. And they're now, the Chinese, the Royal Academy in London, the Europeans and Americans are working together. So we have a joint international consensus on what the rules should be.
Mm-hmm. All right. Let's see. We'll see if we can answer some more questions. We only have 15 minutes, but we have a bunch of questions that are just great questions here. So William Marco, this question is there any common thread that runs through great discoverers or innovators such as Leonardo, Jobs, Einstein, Franklin, and Doudna?
There are 2 common threads in all of my biographies I've discovered after a while. There are more common threads, but the 2 most important of these. One is curiosity. Steve Jobs was not the smartest person in Silicon Valley, but he was very curious. Leonardo da Vinci wasn't the smartest guy in Florence in the early stage Renaissance, but he was curious about everything, about why water swirls when it passes a rock, and why seashells have the same type of swirls mathematically, why the sky is blue. He puts in his notebook, describe the tongue of a woodpecker. I mean, he's just a really passionately curious person. And then I get, when I'm reading Einstein's notebooks, why is the sky blue? That curiosity, we all have that curiosity as kids. Jennifer Doudna collected seashells and tried to study the spirals that happen. She loved sleeping grass in Hawaii. When you touch it, it curls. Now, when we're kids, we're all in our wonder years and we all are curious about these things. But many of us after a while, kind of lose our curiosity. We get told by grown-ups, quit asking so many dumb questions. And so one common thread is that my people that are very creative, meaning my biography subjects, never outgrow their wonder years. Even as they're older, they're still asking, why is the sky blue? What does the tongue of a woodpecker look like? And that basic curiosity, like, why do bacteria have these weird things in them, especially if they grow up in salt pond environments? There's no reason to try to figure that out except for curiosity. And then one day you figure it out, just as we're coming into pandemics, and you say, oh, I'm glad I figured it out because it's useful. What you do, Trivia, and what Tulane, I hope will create a lot of Spencer Olewskis in New Orleans will do, is you move things from bench to bedside. It means you take a basic curiosity-driven discovery that's done at a lab bench, and eventually you find a way to bring it to a patient's bedside and have translational medical uses for it. The other thing that most of my main characters share, all of them share, is that it helps to be interested in all fields of endeavor, whether it's Ben Franklin or Leonardo da Vinci or even Jennifer Doudna. They like music and they like math. They like anatomy. They like astronomy, but they also like art. They like music. They like zoology. And that ability to stand at the intersection of the arts and sciences or the intersection of the humanities and technology, that's what makes Jennifer more important than the other scientists in this book. That's what made Steve Jobs a lot better than the other techie engineers who could code better in Silicon Valley in the 1980s. And that's what made Leonardo da Vinci. That's what Vitruvian Man is a symbol of. The guy, self-portrait of Leonardo, standing there naked in the cosmos on the planet Earth, spiritually connecting art and science. How do we fit in? And Steve Jobs, when he launched a product, it always ended with that slide of the arts and technology street signs. He said, if you stand at that intersection, that's where creativity occurs. So that's what I like about Tulane, and that's what Dean Edwards and the deans of the engineering and science school, others are doing, is trying to get as many possible students, not only to double major, but to double major in disparate fields, to be able to take history and biology, or to be able to double major in things like literature and physics. And that's one of the things that has been a push at Tulane, and I think it creates people who are more imaginative.
Mm-hmm. Well stated. We have another question from Lynette Bates, which is an interesting one. Please share the most surprising revelation which you discovered during the writing of your book.
Well, one thing was how easy CRISPR is, and that you can order off the internet the reagents, the substances you need to do a CRISPR gene editing. There's a character in my book named Josiah Zahner, who you probably won't like, Trivia, but I was affectionate towards him. He's a biohacker. He doesn't play by the rules. He does it in his garage. He uses CRISPR to make super frogs or Dalmatians that glow in the dark. And he even shot himself up with the CRISPR editing that would change the myostatin regulator so he could do greater muscling in his body, which didn't work, by the way. But that notion that this is something that I can do with a couple of graduate students, or Josiah Zahner can do in his lab. And so he's a little bit like Puck in A Midsummer Night's Dream. He pops up, 3 or 4 times in the narrative to sort of play the wise fool, the ones who say, what fools these mortals be, and to remind us that this is something we're going to have to figure out as a society, all of us, how we're going to control. It's not going to just remain the private domain of research scientists.
Yeah. And I agree with you. We have those Josiahs to remind us of the importance, of not only promoting what this could do, but also being, watchful with respect to those applications. We have another question.
Hey, there's one for you. I'm going to interrupt because my friend Julie Orick said, how did Trivia come up with the name of her company?
Great
So
question.
Julie, who I know how her family company was named. It's called Orick, but maybe you could explain Obatala.
Yes, great question. Thank you for that, Julie. Obatala or Obatala in the Western Yoruban language and region is an African Orisha that was tasked with, as the story goes, forming the human body using naturally occurring materials, clay and mud. And so at Obatala Sciences, we use naturally occurring biological materials to mimic the tissues of the human body. And of course, we thought it was fitting to name the company after Obatala, in the sense that we are working to, fill that gap with respect to the tissues that represent various demographics of patients from various backgrounds. Thank you for that question.
I do see also, I'm sorry I'm hijacking, but I see all the people in this chat that I know that I would love to answer the question about, tell us about how Anti-Crispr works and what Doudna is doing. Because everybody says, this is frightening. Terrorists could use it, or Hitler could use it, whatever. I think the major source of funding in Jennifer Doudna's lab at the moment comes from the Defense Department, DARPA, the Defense Research Agency. Partly, so that they could, figure out ways to make sure soldiers exposed to radiation wouldn't develop cancer, that type of thing. But also because Russia, Putin has said, we're going to use CRISPR, to create better soldiers. And who knows, they could even more frighteningly, in some ways, create better mosquitoes that could, spread pathogens and diseases. And so in Doudna's orbit, there's a person who's pictured in the book, and they create what's called Anti-Crispr, which, just like, in a Superman comic or whatever, a Marvel comic, it is what it says, which is, how do we stop CRISPR? If somebody does a type of gene editing, how can we find ways to make it not work? And so that too is an important part of the process.
Yeah. Let's see. We have so many wonderful questions here. There's one from Emma Rogers, who is another Tulane graduate. She graduated from the biomedical engineering program, and she's also one of our recent team members on the science team. We're so excited to have Emma as part of the team. She asks, do you see the most immediate ethical considerations? What do you see as the most immediate ethical considerations as we, as a world, will be forced to reconcile with regarding stem cell and CRISPR technologies?
I'm going to let you take stem cells because that's your field. Although I will say that the editing of stem cells is the way that Victoria Gray a few months ago became the first person cured of sickle cell. That by editing stem cells, we can do a lot in the patient using CRISPR to take on diseases, especially of the blood. Certainly when we get to cancer, we can do T cells and others edited so that our immune system will fight cancer cells, especially very targeted cancers, much better. But the ethical issues, and I think it's the most immediate ones that Emma asked about, not the creating babies with super IQs that'll happen 30 years from now. I guess the most immediate ones would be the safety and off-target issues, because if you make these edits, for example, if you edit out sickle cell, or if you edit out, as the Chinese doctor did, the receptor for HIV, sometimes these things have other uses, like it helps you fight malaria, or it helps you fight West Nile virus. And there are 3 billion base pairs, I think Spencer told me, in the human genome, and they're complicated at times. And when we decide we want to play God and fiddle with some of that, Francis Collins, the head of the NIH I mentioned earlier, he says, we have to have a little bit of humility before we fiddle. So I guess right now, people like David Liu at Harvard are coming up with safer, more targeted ways to do editing. But I think we have to be careful of unintended consequences when we start fiddling with the letters of our genetic code.
Yes, and for the most immediate future, Walter, it is certainly very similar with respect to the stem cell applications and the ethical concerns there. There are, individuals who have taken it upon themselves to use stem cell therapy, and the outcomes have been unfortunately not in the favor of the patients. In one case, in one instance, the patient went blind. The stem cells they will function in the capacity that they are meant to function. And, we don't know how to turn them off when they start going. They are a powerhouse and they are manufacturers, and that's what they do very well. And so there's the fear of the development of tumors. There's a fear of the development of blood cancer and those off-target effects that you're speaking about. And, I think we would love to address all of these wonderful questions that are in the chat room, but we are winding down time, unfortunately.
I tell you what, Trivia, I'm going to do guest prerogative. And if you don't mind, I'm going to do a lightning round of a few I've seen right now. And if people have to sign off, they can.
Yeah.
But real quick, there's some really great questions. Larry Kaiser said, hey, Feng Zhang was the one who first did it by 2.5 weeks, figuring out how to do it in mammalian cells, which was different from what Doudna was doing. Was he bitter about being left out of the Nobel Prize? I thought he might win the Nobel Prize as the third person. I talked to him right afterwards. He said, well, it's sort of a relief now that it's over, but I'm deeply disappointed. Mm-hmm. The patent battle and the Nobel question is the question of obviousness, which, by the way, is a legal term, which is once it was used in a bacterial system, was it obvious that it would work in the human system? Feng Zhang, of course, says, no, it wasn't obvious. I actually think he may be right. But when it comes to the basic science discovery, the Nobel Committee said that goes to Jennifer Doudna and Emmanuel Charpentier. And Sarah, real quick, Kevin said Jennifer and Emmanuel from different disciplines. That's what I'm talking about. It's important to bring different disciplines together. In this case, Emmanuel Charpentier was an expert on small organisms, microbiologist. And she partners with Jennifer, who, as I said, loves structural biology and chemistry. And by putting those two together, you get the fundamental answer. And finally, the Sarah Bitzel question, as a parent, because I think we ought to end this because a lot of people here are parents on this note, we all emphasize making sure our kids knew how to code, i.e., knew C++ or Python, or tried to have their schools teach them computer coding. And now she says, the more I watch and learn about this, I can't help but wonder whether the emphasis should be placed on genetic coding. Mm-hmm. That is the purpose of this book, in a way. I think computer coding is going to be really important. It's going to be part of this new revolution. But those of us who wanted our kids to learn computer coding are now going to have kids who are going to say, yes, but I also need to learn the code of life, because that coding, and it's not necessary that you have to know what ATCG each one is in the human genome, but you have to have a feel for what genes do. But it's not just our kids who have to have a feel for what genes do. The reason I wrote this book is that nature is beautiful, as Trivia said at the beginning. It's important for all of us. I mean, if we're going to say, I don't like GMOs, that's fine. But it's useful to know what a genetically modified organism actually is and what a gene actually is. And if we're going to be part of this discussion with our kids on how we're entering this new era of the life sciences, it's helpful to know what a gene is. It's useful to know how gene editing works. But, and this is important, the basic curiosity is also beautiful. It's wonderful. This is a line I use in the book and Trivia used at the beginning. It's really, there's a joy that comes from understanding how something works, especially when that something is ourselves. So thanks, Trivia, and thanks for letting me go for a minute over.
No problem. Thank you, Walter. What a wonderful way to end an exciting conversation about your book. Go get the book, people. It's awesome. It's amazing. And thank you to everyone who attended the session. Thank you, Walter, for a great conversation. And we'll wrap it up.
Thank you, Trivia, for all you did. And bye-bye.
Bye-bye.